The second part of the characteristics of the genetic code. Now, they say that the genetic code is universal. What do you mean by the word universal? Look at this beautiful picture right behind me. They say that starting from the simplest organism, which is the bacteria, all the way up to complex organisms, which is you and me, the genetic code is universal, or it remains the same, which means that you will have 3 stop codons, whichever organism it is. You will have 61 sense codons, whichever organism it is, and you would also have the first start codon always being AUG. So since all the rules are followed in every living organism, they say that the genetic code is universal. Now there are few exceptions here, and let us see what are those exceptions. Now they say that the mitochondria of yeast, as well as a bacteria by the name of mycoplasma, right, they show slight difference in the understanding of the genetic code. And what is that difference? Let us see one exception here. So you know that the universal code says that UGA is a stop codon. But in case of mitochondria of the yeast as well as mycoplasma, in both these cases, they say that UGA actually codes for tryptophan, indicated by 3-letter code here, TRP. So that's an altered code, right? Otherwise, the genetic code is absolutely universal for every living organism, with few exceptions here and there. Is it okay? Moving on. Now, what is the degeneracy of the genetic code? I just talked about that in my previous module. Let's understand this part little again. So what you see here again is the table of the genetic code, right? And by degeneracy, what was said was many codons can code for the same amino acid. Let us see some examples here. Look at this example down here. What you see here is that GGU, GGC, GGA, and GGG all 4 code for glycine. Let's see one more example here. Look at that. What do you see? GUU, GUC, GUA, and GUG all code for valine. So when 2 or more codons code for the same amino acid, it is called as degeneracy of the genetic code. Or we can also say that the genetic code is degenerate. Is that okay? I'm trying to put a point here. What is that point? Let's see. So now we know that number of sense codons that are there in your genetic code table is how many? 61. You get that right. And number of amino acids are how many? 20. You get that right also. Now for those 61 sense codons, you would roughly estimate how many tRNAs to be there. Ideally speaking, for each codon, there should be 1 tRNA to bind on it, right? So expected number of tRNAs will also be 61. Correct? And why is that? Because a specific anticodon of tRNA has to base pair with a particular codon on the mRNA. Yes? So we would expect that there are roughly about 61 tRNAs moving around in your cell. But remember, there are much less anticodons that you would see in your cell, or much less tRNAs in your cell. Now why is that? Why are there less anticodons, or why are there less tRNAs? To understand this, we will talk about wobble hypothesis in the next module. Okay? So remember that anticodon of tRNA can read 2 or more codons on the mRNA. And how or why is that possible? We'll see that in the next module. So in this module, I have completed the characteristics of genetic code. In the next module, I'll be talking about something called as the wobble hypothesis, which will tell you how a typical anticodon can bind to 2 or more codons during protein synthesis. Thank you and take care.